Thank you, Jess. So can I start? Thanks for the introduction. My name is Masashi. I'm based in Oxford, working for Oxentia as an innovation consultant from the Oxford University. Plus, I run a company called Biospire. And I'm happy to support this event because I am an official ambassador of LinkJ, based in the UK. And it's like a three years continuous the planning and ongoing for this innovation workshop. Thanks for the organization by the One Nucleus team. And before COVID, LinkJ did put in a physical booth. And Aki-san, for example, like a CEO of LinkJ, did a speech. But it looks like many years ago. And from this year, we're going to restart. Tomorrow, I will be in London with standing in front of a LinkJ stand. So I want to see you all. And yeah, in fact, the connections between Japan and UK, thanks to the Brexit or not, it's increasing, and the government are increasing for like engaging FTA, etc. And the ecosystem in between are also increasing in both in the business to business or startup to industries. Also, we have to increase more like opportunity with the investors to startups. So this is a kind of an opportunity that's we are trying to increase more engagement. So this is opportunities for this 1 hour session between the Japanese startups and not only, but global stakeholders like you. Thanks for joining for the audiences. So before starting the brilliant presentations, maybe I hand over to Aki-san, CEO of LinkJ, for the brief introduction of LinkJ.
Okay, thank you very much, Masashi-san, for your fantastic introduction. So, hello everybody, good morning everybody in London, UK, and Europe. Yeah, I'm Aki of President and CEO of LinkJ. So today we have this timeline. So I'll make a short opening remarks. Then after that, three companies will make a great presentation. Then closing remarks, Masashi-san by Masashi-san. So at first, I would like to share the overview of LinkJ. So LinkJ is a non-profit organization, and its official name is Life Science Innovation Network Japan. L, in a wide range of life science. I, to support and promote innovation. N and K, by creating network or ecosystem. J, both in Japan and abroad. That is our objectives. We were established about 7 years ago, and we are a membership organization with more than 600 members at this moment. And for example, we are hosting a lot of symposium seminars and also hosting acceleration incubation programs to support the innovation. So for the last 1 year, LinkJ as well as LinkJ members hosted more than 500 those symposiums, seminars, and programs. So
sorry, Soyama-san.
Yeah.
Your slide is not moving on the full screen.
Yeah. Azusa-san, how about? It's not moving. Maybe, yeah, my staff is now managing it. So I'll continue for the time's sake. Is that okay?
All right.
So we are located in the very center of Tokyo. The town's name is Nihonbashi. It's only 10 minutes walk from Tokyo Station, and with a heavy concentration of Japanese, especially pharmaceutical companies like Takeda, Chugairo, Astra, Daiichi Sankyo, and so on, for the last 300 years or so. And within in this town, LinkJ's facility is located in 15 buildings, and those include big conference room, small meeting rooms, co-working spaces, and also wet laboratory for startups to share with biosafety level 1 or 2. So those are our overview of LinkJ. And today, three very prominent and promising startups will make a presentation, and I hope everybody here will enjoy it. Thank you very much.
Thank you, Aki-san. Yes, and for the audiences, if you're interested in Japan business development, you have to have an office in Exchange site.
Yeah, I love that.
Great, thanks for the introduction. So go to the technical session. So first speaker is the Pepti Growth, and I invite Robert Brownleet. And Pepti Growth is, I believe this is like a game-changing technologies. And if you are in the regenerative medicine field or cell therapy field, you may be struggling with some growth factors or cytokines. And Robert will go details, but I believe I love these technologies, and this will help. This should help for like a scalability, etc. Because it's quite booming for cell therapy fields. Plus, for example, like a cultivated meat, which you need kilos of final product. So Robert is an experienced person on this business. So maybe I hand over to Robert.
Sure, thank you, Masashi.
Thank you.
Can everyone hear me okay?
Yeah, perfect.
Well, it's a pleasure to meet you all on this rainy morning in the UK. But I was very pleased to come on board and present about PeptiGrowth. My background is in recombinant proteins. And PeptiGrowth, really, when I heard about PeptiGrowth and what they were doing, I was very interested to come and work with them, because they're providing a really innovative solution to issues of scale and cost that are present when you talk about recombinant proteins at the moment. So without further ado, let me share my screen, and I will share the presentation. Now, can everyone see that okay?
Yep, go ahead, thank you.
Good. So PeptiGrowth are providing peptide alternatives, as Masashi said, to conventional growth factors. And usually growth factors in terms of cytokines come under the form of recombinant proteins. And they're becoming very, very important for cell therapy and regenerative medicine. And in addition, as Masashi mentioned, cultured meat. So from a sustainability perspective, there's big drives to develop cellular agriculture in order to lower the CO2 input and provide sustainability for the planet. So let me move on here. So who are PeptiGrowth? Well, PeptiGrowth were founded in 2020, so they're a relatively young company, and they're a subsidiary of Mitsubishi and PeptiDream. Now the Japanese members of the audience will be familiar with PeptiDream. It's one of the biggest biotech companies in Japan. And they are involved in developing, manufacturing, and marketing of alternative peptides with equivalent function to conventional growth factors and cytokines. And the applications are in cell therapy and regenerative medicine currently. And the big feature about the PeptiGrowth products is they're completely produced through chemical synthesis. And for those who are aware, recombinant proteins are usually grown in culture using a prokaryotic or eukaryotic vector. Often the serum is used in the manufacture, and there's issues of biological contaminants. So whenever you're dealing with vectors, you've got potential endotoxin present, which are pro-inflammatory. There's also issues in terms of quality and reproducibility in terms of lot-to-lot variability, and they're not that stable. So PeptiGrowth have solutions to all that, and of course the big thing is they enable xeno-free, so totally animal-free manufacture, for chemically defined cell culture media. And PeptiGrowth have developed a platform called their Peptide Discovery Platform System, and basically they have 800 plus building blocks, and which is expanding so far in excess of your standard 20 amino acids. And you pick 16 or 24 building blocks based on the protein that you're trying to mimic. And these building blocks are randomly positioned to form a diversified peptide library. And then they add a readable and amplifiable tag. And so if you work it out, 10 random positions with each position possible of being in any 20 of the building blocks, there's over 1 trillion combinations there in terms of peptide manufacture. And once this peptide library is developed, it's then screened in vitro for target binding and activation. So in terms of conventional products at the moment, now we mentioned recombinant proteins. Obviously, FBS is the gold standard for cell culture currently. Now, there's a lot of issues with FBS, and the market as a whole is trying to move away from bovine serum. namely because the homogeneity is poor. Every lot is different. That contains a lot of biological impurities, antibodies, prions, bovine viral diarrhea, etc. And the stability is short, so it's not very stable. Now, there's one solution that has been proposed and is actively being used for the market as a whole is recombinant growth factors. Now, this is, although the technology is very interesting, you know, they genetically modify microbials, and these proteins are then expressed in vectors in bioreactors. And you can get functioning growth factors through after extraction and purification. Now, again, the homogeneity is better than FBS, but there's still issues with lot-to-lot variation. Some growth factors, the yield is low as well, so you know, there's issues associated with that. And obviously they're biotic in nature, so you're still using either a bacterial or eukaryotic vector, and the stability once reconstituted is short, particularly for growth factors such as FGF. They have to change the culture media regularly because it's notoriously unstable. So the peptide alternative solution is completely chemical synthesis, and as a result, the homogeneity and lot-to-lot variation is spot on. So it pretty much eliminates lot-to-lot variation. It's completely abiotic in nature, and because the proteins are cyclical in structure, they're a lot more stable. So again, that they will be much stable, not closed vial, and so they'll last longer when refrigerated and frozen, but also open vial. The stability is much better, which is what the market needs at the moment. So in terms of the products, they've, in a relatively short amount of time, they've already launched 4 products in terms of HGF alternative peptide, TGF beta inhibitor peptide, a BDNF alternative, and a noggin-like peptide. And they're going to be, they've got over 20 products identified, and they're going to be releasing those. Aim is about a product per quarter for the next period of time. So this is the HGF alternative peptide. And we know HGF is a c-Met agonist, so HGF binds to the c-Met receptor, inducing dimerization and exerting its biological activity. PGO1 binds in a similar fashion to the cellular receptor, and again induces phosphorylation. Here you have some data. So this is in terms of c-Met phosphorylation, your percentage activation on the y-axis, and you can see HGF compared to PGO1, and you can see the equivalency there in terms of c-Met phosphorylation. This is in epithelial cells, so HUVEC proliferation, and you can see HGF compared to PGO1 in terms of activation and the equivalency there. Now, bear in mind that PGO1 is fully chemically synthesized, so the molecular weight is about 1/16 that of HGF. So you need a lot less of it in order to get the equivalent activation. It comes as a lyophilized protein stored at -20, and this is an example of the vial there. Now, this is some data that was carried out by Professor Taniguchi at the University of Tokyo, who's a leading expert in the field. He carried out a proliferation assay of human liver cells. And you can see that the cells were enhanced by the supplementation of PGO1 and compared with HGF. And this is your HGF negative in terms of proliferation rate. This is standard HGF, and this is the PGO1 protein. You can see the equivalency there. He also carried out a cloning assay in iPSC-derived liver cells. And you can see in terms of cell number, the equivalency with HGF and PGO1. And also, interestingly, from a clone perspective, you can see the HGF clone number there. And there was an increased clone number formation in terms of using PGO1. Now, TGF-beta inhibitor is widely used in the field, and there's 2 forms of TGF-beta1 inhibition currently. And you can use a small molecule kinase inhibitor, which is an intracellular inhibitor, so there's issues with specificity there. And you can also use an antibody, which binds to free TGF-beta1. And PGO2 binds to free TGF-beta1 and has a similar method of action to the antibody. And here's equivalency data. You can see an SBE reporter assay comparing PGO2 and anti-TGF-beta1 antibody, and the percentage inhibition is comparable there. Now I'm conscious of time, so I'll pick up the pace a little bit. So here you have data on the BDNF alternative. And we know BDNF binds to the TrkB receptor, inducing dimerization. Very important for neurological organoid modeling and neuronal studies. The PGO3 again binds to the TrkB receptor, inducing phosphorylation in a similar manner. Here you have data in terms of TrkB phosphorylation and percentage activity of BDNF versus PGO3. And you can also see the reporter gene activation. Interestingly, there's a slightly lower, higher activity and a lower concentration in terms of reporter gene activation. And again, from a molecular weight perspective, because the peptide growth peptides are sequentially different and contain just the active portion of the protein, they're a lot smaller, and this is less than half the molecular weight of standard BDNF. So you need less than half of the actual peptide. Noggin-like receptor, again, very, very important, very difficult to make in recombinant protein cell culture. Noggin binds to BMP4 and BMP7, and PGO4 binds similarly to free BMP4 and BMP7. Here you have equivalency data in terms of percentage inhibition of BMP4 in comparison with Noggin, and here you have the percentage inhibition, of BMP7. Now we have an independent inhibitor for BMP4 coming shortly, and also an independent inhibitor of BMP7 as well. And this is the pipeline. So as you can see, there's a very comprehensive pipeline of products which are coming, including some very important growth factors. Wnt3a, R-spondin, notoriously difficult to make recombinant proteins. They're very expensive, low yield. HGF, FGF, activin A, all used in very important growth factors used in industry, including the interleukins as well. Now, what's really an important point to make as well when it comes to the peptide growth solution is the scale. Now again, in recombinant protein manufacture, you can really, in a 75-liter grow bag, you're making grams of recombinant protein. And Pepti Growth are partnering with PeptiStar, who are CDMO, GMP, fully GMP CDMO, and they can make, in their R&D building, they can make 20 grams of these growth factors in their smallest 1-liter autosynthesizer. In their 10-liter autosynthesizer, and bear in mind this is their R&D building, they can make 200 grams. So far surpassing the scale of recombinant protein manufacture. When we go into their manufacturing 1 building, we can make 500 grams per batch, and even in terms of the record for manufacturing 1 building is 1 kilogram per batch. So it really provides a whole host of opportunities in terms of large volume manufacture at a low cost for the market. And when you talk about the big building, the manufacturing 2 building, they can make 5 kilograms per batch of protein. So it hopefully will provide the industry with a scalability never before seen for the market globally. And that's everything for my presentation. Sorry, I ran over a little bit, Masashi, so apologies for that.
No, that's okay, no problem. Thank you very much, Robert, for the great presentation. If you got any questions from audiences, you can just unmute. We got allowed 3 minutes for this Q&A, or you can type on the chat box. Before somebody raises hand, maybe I have a question to Robert. So you are from not competitive, like a conventional the growth factor field. And can you explain what's your role in the European UK market? Plus, what's your honest think of this product discovery?
So again, I used to work for a recombinant protein manufacturer. So my background, of technical background, microbiology and biochemistry, and I've been in the business side of the industry for about over 20 years now. And I was involved in European recombinant protein business. And I came across Pepti Growth and was really wowed by the technology. So I was brought on board as a consultant business director for Pepti Growth in Europe. In order to help promote really the Pepti Growth solution. And my feeling is that it really particularly for areas where big bioreactors are looking to produce big quantities of tissues. So not just cells but tissues. So such as the cultured meat market, cultured leather. It's one of the largest growing markets globally at the moment because it's very important from a sustainability perspective. This is just the perfect solution, and it's really, really interesting that this technology is out there. And when you look at CAR-T manufacturing, cell therapy and the applications in terms of, I mean, there's big biotech and pharmaceutical companies that have basically got massive cellular manufacturing plants in development with a view towards manufacturing either proteins or cells, so expressing proteins or cells for regenerative medicine and cell therapy. The scale is, if we're going to roll out these therapies globally, is really, really important. And recombinant proteins just don't have a solution at the moment. They're cost inhibitive and scale inhibitive. So particularly for CAR-T, I think there's big investment going in there. Again, that's a high growing industry, and it's going to be really, really interesting to be able to provide the world with these treatments, utilizing lower cost and scalable solutions like that of PeptiGrowth. So very exciting, and I think it's super interesting.
Yeah, thank you very much. Because I was from Cambridge stem cell company, and was struggling as doing operation, struggling for the stability and like a lot of batch to batch differences, etc. So I wanted to have like this product beforehand. But anyway, Robert, thank you very much for your presentation. That's great kickoff. And for the time strains, maybe we can go for the second speakers. We invite Hideaki Sato from Luxana Biotech. And his company is doing a nucleic acid drug discovery, and which is, I believe, a high growth market. And the company is coming from University of Osaka. And this is a great opportunity to be in global sessions to have a further engagement with global stakeholders. So all for Sato-san. Thank you.
Okay, thank you for the great introductions. I'm Hideaki Sato from Luxana Biotech, President and CEO. So this is a great time to introduce our technology and go up to the collaboration opportunities. So we have the great technology for the realized nucleic acid drugs. So utilize the artificial nucleic acid called XNA. So these kind of technologies brings right to the patients, we believe. So we are located in Osaka. In spite of the located in Osaka, LinkJ friendly invited to join the memberships. So we are 5 years old companies, and so business started was February 2018. So me, Hideaki Sato, and Professor Satoshi Obika from Osaka University, he is a co-founder of the companies. So he is a pharmacologist and very special, deep knowledge for the nucleic acid chemistries. So we are in the location in the inside of the Osaka University incubation unit, and we next steps we go out to the this is honestly says the Osaka University is very helpful, but we are going to the outside of the Osaka universities. So we are currently we are 27 people working with me, and equity history. We do from the seed round to the Series B. The total is 1,970 million JPY. So the nucleic acid drugs is a promising drug modalities, we believe. So we understand nucleic acid drugs is a long history for the from the first invention to the realized to the actual drugs, like from the Ionis and Alnylam. So and this is a two giant needs 20 to 30 years for the realized drugs. But their effort is a very great steps. The nucleic acid drugs is realized to the market. So the still position is not so big size, but we very expected the sales growth is better than compared to another conventional small molecules. So we are now focused for the nucleic acid drugs realized. So further, the challenge is realizing nucleic acid drugs. So key issue is to overcome the two toxicity. So the generally, nucleic acid drugs is a focus for the systemic genetic disorders and or the central nervous systems, CNS areas. And this is a two promising area drug target for the nucleic acid drugs. So for the systemic administration, sometimes occurs serial hepatotoxicity and adverse event occurs. So that we should control the administration volumes and volume of the drugs. On the contrary, the intrathecal administration, this is a very succeeded for the Spinraza from the Biogen's drugs. So these drugs is a directly intrathecally administration to go up to the CNS areas. This is a simple route and maximum efficacy is coming to by directly administration. But sometimes occurs heavily acute neurotoxicity and delayed neurotoxicity. So if toxicity occurs, the dose cannot be increased, maximum efficacy cannot be obtained. So that our 5 years experiment, so we focus on how to manage the two toxicity. So our core technology is 4 different kind of nucleoside. So you can see the right side left side. So first generation 2' MOE, Spinraza is a very succeeded drugs. But this kind of structures need to the more knockdown activity, need to the binding affinities still needed. So that over the 20 years ago, second generation LNA, they call the locked nucleic acid. Locked means blue lines, so locked. So this kind of lock given the highly potential of the affinity to the target RNAs. So but these kind of structures also occurs heavy hepatotoxicity and still needs a lack of the biological stabilities. So that these issues still remain some innovation needed. So that our Professor Obika's his group finds a different kind of the bridge type nucleic acid, locked nucleic acid structure. So Japanese says the bridged nucleic acid structures are a little bit different pronunciation, but tendency is the same. So keep the highly affinity and how to manage and reduce the hepatotoxicity and neurotoxicity and stable in biomatrix. So these kind of bridge type structures so limited to the usage. So that Professor Obika's groups find another kind of DNA, a simple DNA structure with scleropropylen structures. So these structures given the highly stability for the DNA without phosphorylation. Phosphorylation is one of the big issue for the toxicity. So this is a mice model data. So two toxicity can manage by our XNA technologies. So you can see the right and left panels. So RNA gap, this is one of the structure of the nucleic acid drugs. So this structure is given the wing portion, the green circles. Now this is a modified point, and we simply change from RNA to amina and RNA from RNA to amina and SCP, and this is the structures. You can see the RNA original structure is given the highly hepatotoxicities, and if we when we change the wing portions, so you can find you can see the significant reduction for hepatotoxicity, so showed by the AST levels, the drastically reduced. Even if this administration is 30 milligram per kilogram, so heavily duplicated administration cases, even in these cases, we can manage to minimize the hepatotoxicity. On the right panel, you can see the neurotoxic RNA gap mouse. This antisense giving the heavily survival rate of infection during the ICV, and ICV injection is directly to the administered to the brain. So this kind of studies is very severely occurs survival rate and behaviors. So now in these cases, we substitute the DNA portion, the pink circles, the change to the 5' CP, and so this modification can give it reduce the neurotoxicity. So we have not only these data, we have the mice model data and non-human primate model studies. We observed the good structure, this structure given the good technology, these good character. So based on these technologies, we have one is the core technologies. One is excellent nucleic acid chemicals, we call it XNA, and integrated antisense drug discovery platform, LuxSeq. So these two technology cores, given the XNA technology, are licensed gene by gene models and collaborative drug development with pharmaceutical companies, and in-house original drug development are licensed at IND-enabled stage. So based on this technology, we have good succeeded in creating multiple pipelines during the 5 years. So now we are very regret to the all of the partner cannot disclose current state, but we have the 5 different kind of pharmaceutical company or the chemical companies for the multiple target, and this is a collaborative development works. And XN Tech out license succeeded to the Arigos Therapeutics and Takeda Pharmaceuticals. And in-house original drug development, we are currently very focused for the subacute phase spinal cord injury treatment by antisenses. So you can see that this kind of collaborations, genetic disorder and CNS area, so that our technology given such kind of the new areas, giving the very effective point. So our currently business area is screening to the non-clinical to INDs, because our core asset is very suitable for the screening to the candidate nomination, and clinical stages. Currently, we need help for the pharmaceutical companies, so that we focused for the R&D area, this area. So our management team is most of people coming from the pharmaceutical company and biotech companies. So unfortunately, all of members are coming from Japan, Japanese people. So, but we are open to not only Japanese people, so global talent is also welcome to join to our companies. So our currently the new drug development has been launched 2 years, two projects per year, and most advanced project in the preclinical stage is spinal cord injury treatment. And calendar year 2030, so we are very promising to come up to the late stage clinical stages and giving the much better efficacy coming. So now we are looking for the three kind of the partnerships. One is finance, is the Series C investor, calendar year 2023 Q2 Q3 we are currently the target. And project investment is welcome to the token. And pharmaceutical companies now still we are looking for the good partnership, the genetic disorder and CNS. And tech tools is one is a key point is a DDS tools. So but we some indication not needed to the DDS tools. And AI and machine learning is the next challenge for our technology go up to the next stage. So we really hope to realize the innovative drug development with the power of the nucleic acid chemistry and contribute to the patient and supporters. So that thank you for the pay attention.
Thank you very much, Sato-san. I got 1 or 2 minutes. So if you got any questions, put in a chat box. But if I can crack on, so your pipelines looks be more like a Japanese focused, and having one exception, Aligos, in the US. Yeah. What is your strategy for the European development?
Yes, currently so we are just now talking to the European people, European pharmaceutical companies. So before that, we don't have any representative in Europe and something. But this year, next year, we trying to the Bio Europe or something. So this is the first challenge, and we started some pharmaceutical company to negotiate the CNS. CNS is one of the key points for our technology go up to the European companies collaboration.
Sure. So the last slide, you go like a 3 pies. One is financial, the pharma, and the tech, and the pharma collaboration is the priority for you. And the business model will be mainly licensed out, not for you to do clinical study by yourself.
Yes. So this is one big challenge for the spinal cord injury treatment. Is currently we will go up to the non-human primate model studies. So that next early summer is one of the key points for the great sales.
Great. Thank you very much. Thank you very much. Okay, thank you. If the audience is interested in collaboration, just let me know. I will connect them. Thank you very much, Sato-san. All right, thank you. I like doing this session with Japanese, with Robert. People are keeping time on time. Many thanks. And the last speaker is the Masaya Hagiwara from RIKEN. And he is talking about, I think, more than the organ chip, so the called the CubeIn chip, and I think MPS organ chip is quite advanced in European communities. I see many like productions coming from European companies. I don't specify the names, but fundamental technologies are happening in Japan as well, and especially RIKEN is a leading national institute. So I think as Hagiwara-san will explain the basic technologies and looking for the further collaborations. So all for Hagiwara-san.
Yeah, thank you very much for kind introduction, and thank you for giving me this great opportunity to present our research. Actually, we are not the initiating company yet, but I hope we can find a collaboration to initiate a company very soon. And I'm Masaya Hagiwara from RIKEN Japan, and today's talk will be about how we can improve organoid development and integrate with the MPS or organ chip to achieve more sophisticated model in vitro. So let me start from the background. In preclinical study, both of the in vitro and the in vivo model will be used, while the both models have pros and cons. However, the accuracy of those models is never sufficient, and it causes huge cost and time for drug development. In order to counter this, new in vitro experimental models are rapidly developing in the world. Organoid can express the complex biosystem by establishing mini organs from stem cells, while organ-on-a-chip or MPS replicate the circulation flow in the microfluidic chip to express tissue-tissue interaction. The final goal of both technologies are same, but there are no suitable integration of both technologies, and they still have many challenges. In organoid technology, various types of mini organs have been developed in the past 10 years. However, there is still a huge gap in morphology between organoid and actual organs. This is largely associated with the differences between the culture environment in the lab versus in the body. The current prevailing organoids are mainly spherical shaped like these. And there are no inlet of drugs and outlet of metabolized substrate. Without taking care of the proper morphology, it is quite difficult to use them practically for drug screening. On the other hand, MPS or organ-on-a-chip also faces in difficulties, especially when it comes to use organoid. One of the major bottlenecks of organ-on-a-chip is the system complexity. The multiple cells or organoids are needed in the small chips, but it makes experimental time increase and hard to maintain cell viability. However, without achieving the good viability from all cells, the experiment will be failed as a system. As a result, only trained operator can be conducted. Also, it takes few weeks or months to develop organoids, but it is quite difficult to culture them in a small chip for long. Those operational difficulty, as well as sample collection and imaging and variation of the experiment, restrict the practical usage of the organ-on-a-chip with organoids. In order to counter these difficulties, we have developed this cubic culture device. On the cubic polycarbonate frame in this yellow line, the agarose is adhered at the wall, and inside this culture container, the culture gel, such as collagen and matrigel, are injected to culture cells or tissue. Agarose wall allows the nutrition pass through and reach to the biosample easily. Therefore, when we want to culture the sample inside the cube, we just need to immerse the cube into the medium like this. Then we can get the same result as regular 3D culture. On the other hand, agarose has enough rigidity to sustain ECM and biosample. Then we can pick up the sample with ECM during culturing and transfer the container and rotate it. As shown here, the handling ability of the sample can be enhanced significantly by using the cube. By taking advantage of this handling ability of the cube, we can use it as a carrier to handle organoids and integrate multiple engineering technology to achieve spatial control of cell and ECM, morphogen gradient in the cube. 4D imaging by rotating the cube continuously and scanning from 3 orthogonal planes, and multi-organ interaction. So today, let me briefly explain for some of the technology due to the time limitation. So starting from morphogen gradient control by integration of the cube and fluidic chip. One of the great advantages of using cube as a carrier is that it facilitates the usage of fluidic chip quite a lot. All you need to do is pick the organoid when it's ready and insert it into the appropriate place in the chip. Then we can start the experiment with the chip. One of the major advantages of employing fluidic chip is the generation of the morphologic gradient, which is a very important factor of organ development. So now we propose the organoid workflow with Cube platform. The Cube allows us to culture sample in a well plate until the timing requiring gradient. But the organoids are not always the same quality each time. In those cases, you can select the sample with only good conditions to insert into the chip to minimize the waste. Then culture with gradient in the chip, few days or weeks to provide the body axis information to the cell. After finishing gradient, take out the organoid with Cube from chip, and you can section the organoid together with the Cube frame so that we don't lose the orientation of the gradient during post-experimental analysis. As shown here, the Cube supplies a consistent workflow from the organoid culture to imaging. This is one of the demonstrations in which we cultured iPSC spheroid in the Cube and supply the differentiation factor gradient from the neuroectoderm and the mesoderm. You can clearly see the localization of the SOX2 and the brachyury, the expression which are the ectoderm and mesoderm marker respectively. Okay, now I will introduce the cell and ECM spatial control using this Cube platform. We have developed this Cube multi-cube device that allow us to place the ECM in our desired location. The L-shaped frame, the frame structure in the Cube enhances the surface tension between the frame and the hydrogel so that the liquid phase hydrogel can be self-standing and localized into each individual Cube unit. The gel are in contact with each other, but thanks to the high viscosity of the hydrogel, only minimal mixing will be generated before curing. Then we can put the hydrogel, multiple hydrogel into the incubator to cure it together so that there is no physical boundary for cell, but we can place a different composition of the hydrogel or growth factor with the hydrogel or the cell types. So we can design and localize the individual component and structure surrounding cells. The multi-cube is quite simple to use. The cube is first placed in the holder for easy handling, then inject a hydrogel with a pipette into each unit before curing. As you can see, the liquid phase hydrogel can be localized in each unit before curing, but do not mix with each other, thanks to the high viscosity. This simple device can be applicable any hydrogel, even very diluted matrigel to control. Thus, many kinds of hydrogel cell types or growth factor in ECM can be localized. As a demonstration, we have conducted heparin binding EGF localization in matrigel. Heparin part of the HB EGF adhere to the heparin sulfate in the matrigel and support the localization of the EGF. When we place a matrigel with the HB EGF in the right unit only, and matrigel without any additional supplement in the other unit, and culture the normal human bronchial epithelial cell like this, the branches will be developed in the right unit only, and any other area is just a tubular shape. So by employing this kind of the localization of the different environment, we can supply the body axis and also the asymmetry condition of the organoid. So by employing this cube, by employing these ECM localization, as well as the morphogen gradient mentioned previously, we can design the developed hybrid model with continuous linkage from MPS to the organoid. so that the drug inlet or outlet becomes obvious for practical usage. So last topic is multi-organ interaction in the chip. Using the cube, we can modularize the organoid and assemble them in the chip to customize the organ-organ interaction for drug testing. By employing this concept, we have developed the BBB blood-brain barrier model in the chip. One side of the cube has a brain endothelial cell barrier supported by the astrocyte and pericyte, and the other cube has a neuron spheroid to represent the brain site. Then, by throwing drug candidate in the blood side of the channel, we can analyze how much test compound can pass through the BBB barrier and reach to the brain side, and how it affects to the brain. This kind of tissue or organ-organ interaction we can easily assemble and monitor and analyze for drug testing. So let me summarize this my talk. The technology here is quite simple, but because of this simplicity, we can integrate the multiple other technology to control the microenvironment. Besides the operation presented here, operation here are quite simple, and I believe it can be used a variety of the researchers and the companies to develop the test screening. Currently, we are aiming at developing the startup company to provide a custom design or standard chip to pharmaceutical company or cosmetic, food, or commissioned companies. We are now working for the BBB as showing here in this presentation, and also the blood retina barrier and the skins as showing like this. And for future work, currently we are targeting on the pharmacology models and the safety exam as well. And the patent, we are currently applying the 7-8 patent, and many of them are started approved in the US and Europe. And currently, we are not starting initiate a company yet, but we have a lot of collaborators in academia side to develop the multiple organoid. And eventually, we are aiming to the linkage of the organoid by designing the morphology of the individual organ and make a linkage in the chip. And thank you for the attention.
Thank you, Hagioara-san. It's great presentation, great technologies. I love that. My question is for the connection to the startup company, because for the organ-on-chip or MPS, somebody have to make device. Somebody have to lead to the end. Otherwise, pharmaceutical company will not just enjoy as a beauty of science. So what's your plan? Are you in a RIKEN is a national institute, but encouraging startup creation by yourself? What is your future business models? You wanna
actually, yeah. RIKEN itself is supporting us for initiating company, and some of the patent belong to my previous institute, which is Osaka Prefecture University, and they are also supporting a lot actually. And we all together trying to initiate a new startup company. And for the manufacturing, currently Cube itself is already licensing to the manufacturer, and the chip has a lot of company to develop. So that what we are trying to do is develop the new 3D or organoid protocol and in the chip and designed it as demand, depending on the demand from the pharmaceutical company, cosmetic and food, the companies from customers, and develop the protocol and put in the cube as ready to use and ship it. Or also we are planning to receive the test compound and test it in our company. And then the data we supply to the users. That's our business plan so far.
That's great. Great comment. Thank you. Maybe my comment or no advice, but Europe is quite advanced. So that's in the US. For example, in a one-on-chip, I see like a CN Bio or TissueUse or Emulate. They are on the Europe and quite strong initiatives to develop the final product. that means a bit of less compatibility in the end. But they have to have like a very quite strong initiatives, leadership to lead the new society, new ecosystem. So maybe Europe is one of the recommended place to do collaboration.
Okay, thank you very much. Yeah.
All right, thank you. Yes, good. Thank you very much for your presentation, and thank you all for the 3 speakers. And this is 3 minutes before. I love the Japanese the sessions. How shall we go? Maybe Aki-san or Jasmine. You are in the first time in the Japanese session. If you have any comments.
I'm on mute. No, I just wanted to say thank you for giving us an insight to Japanese life sciences. It's certainly something new for me today. If anybody has got any comments or questions, please feel free to pop them in the chat box. Happy to feed them in to our panelists.
Cool. I also put my email address. So any questions or any comments, free to send me. I will send over to the speakers, plus LinkJ, plus OneNucleus. Aki-san.
Okay, I have one word. I'll be very quick. So I believe that audience today was very impressed by the sciences, technologies of each companies, and also in Japan there are lots of other various kind of high level science and technologies. But for those technologies and science to become successful, globalization is the key or critical. But Japanese companies or Japanese selves cannot do it very well. So we need your help or collaboration. Thank you very much.
Of course. Good. Thank you very much, Aki-san. So yes, I have the final same words that collaboration is needed, and finally the Japanese government encouraged for the globalization, and so does speaking to British embassies. And after the Brexit or no, they are more open for collaboration to Japan, bringing innovative technologies. Maybe UK, Europe can bring to the further end with having a bit of investment, etc. So this is something that I'm supporting for my life work. And yes, and again, tomorrow I will be in OneNucleus Genesis session in London. So if you're physically there, I will see tomorrow. And thanks very much for the audiences. And I hand over to Jess for the closing.
Thank you. I would just like to say a big thank you to LinkJ for putting this session together, and for Masashi for moderating such a great session, and all of our wonderful speakers, of course. And thank you to those of you that joined this morning. Our next session starts at 11:30, so you've got time to grab a quick coffee. And that will be for our innovation showcase, and we'll be joined by 3 companies that will be presenting. So we hope to see you there, and of course grab Masashi tomorrow if you will be attending in London. Thank you for joining, and I hope you enjoy the rest of the conference.
Cool. Thank you.
Okay, thank you.
Bye bye. Thank you. It was great.